Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem. So today we will discuss about the glycogenesis, the synthesis of glycogen. It means the glycogen is the animal starch, which that is the highly molecular density, and that's show the red color in the iodine solution. So here is the sugar which that's eat or drink in the form of juice, which that is made up of fructose as a simple sugar, glucose to galactose making a lactose in the form of milk, and glucose simple is in the form of D-glucose, and sucrose it means the sugar cane. So this formula shows that the different type of sugar when we will eat the sugar which that's convert into the glycogen for the decreasing the water availability. So our body required less water, so the glycogen is required the less water. So here is the light will perceive and carbon dioxide use, and light will fall onto the thylakoid membrane of the chloroplast in the plant to produce sugar in the form of Calvin cycle, which that run to fix the carbon dioxide and produce sugar. And this sugar after the chemical synthesis, that's a starch synthase enzyme used to make a starch. So the starch is different type of starch, amylose and amylopectin. So here is the different product of the starch, which that's a naturally and processed food is sugarcane, bread, grains, wheat, and burger that is made up of amylose and amylopectin. So here I'll draw a person which that's contain a digestive system. When, for example, sucrose, amylose, amylopectin or starch will enter into our mouth, and over cavity contain sodium bicarbonate. Sodium bicarbonate secretion from the gland is known as the salivary gland, which that creates uh, saliva and amylase enzyme. These partially break down the large polypeptide into the smaller polypeptide and oligosaccharide that go into the stomach, and the stomach will not hydrolyze it, and after the pancreas will produce. Here is the pancreas which contain pancreatic amylase and which that is different type of enzyme amylopectin digestion and pancreatic amylase enzyme which hydrolyze the more bond, and that's convert into the disaccharide, trisaccharide and oligosaccharide. These will go into the lumen that is present is here is the lumen of the duodenum, which contain a enterocyte is also known as brush border cell. So here is the sugar will enter into the lumen when duodenum beside the pancreas is here, and that's hydrolyzed by the enzyme which that is also known as brush border enzyme. These enzymes sucrose hydrolyzed to by the sucrase enzyme are present into the brush border cell, and lactose which that is hydrolyzed to simple sugar in the form of lactase enzyme, and maltase is the maltose digestion. So these will be converted into the simple sugar, and their simple sugar will be actively transported into the enterocyte by the GLUT1 channel and GLUT5 channel. Then the basically fructose will enter by the two sodium ion move inside to one molecule of fructose will go inside, and that's further go into the bloodstream by the actively transport. How it's possible? Let's talk about. So ATP will be used to 3 sodium molecule will be go into the interstitial space of the enterocyte and between the blood vessels, and potassium will be move into the enterocyte to go the glucose into the bloodstream. So here is a bloodstream and GLUT5 channel which that allow the same mechanism which allow to move glucose into the bloodstream. So here is the bloodstream, and that is the move inside by the choosing of ATP is fructose and glucose. These fructose and glucose will be used into the muscle tissue. So this muscle tissue remain into the bloodstream for normalize the isoglycemic level. It means the hyperglycemia is the too much sugar and hypoglycemia is the less sugar. So we will talk about the biosynthesis of glycogen is known as the glycogenesis. So here is the pancreas which that contain a islets of Langerhans, beta cell, alpha cell and delta cell, which that is in the form of group, and is the known as the acini, and this is the basically endocrine hormone but also produce the pancreas the exocrine hormone into the duodenum, which that is the pancreatic lipase and pancreatic amylase enzyme and chymotrypsin and trypsinogen, which that is active by the enzyme is the present in the enterocyte. So here is the liver and muscle which that have a receptor, and this receptor is used for the insulin release from the beta cell of the pancreas to bind or growth hormone which that interact into the insulin like growth factor 1 which that produce into the liver, and there's binding to stimulate the sugar move inside instead of the um, insulin. So that's move inside by the ATPase pump to GLUT4 channel, and 2 sodium ion will move inside and 2 potassium ion move outside. 1 potassium ion will move outside, and these will be produced glycogenesis in the form of glycogen. Either glycogenolysis is also produced or glycolysis, which that's convert the glucose into the pyruvate, and this pyruvate go into the Krebs cycle by the fixation of acetyl CoA conversion uh, to into the citric acid cycle to produce energy, or release from the mitochondria acetyl CoA to produce fats by the using of hydroxymethyl glutarate CoA enzyme to convert the malonyl, and this malonyl will be converted into the fats. So that is the fat will be conversion. Malonyl convert into the fats, amino acid. So this is known as, if fat store is known as a lipolysis or lipogenesis. If lipogenesis means the fat production, lipolysis means the fat reduction or fat utilization. So that's all thing is the about the metabolism, and this is known as the anabolism. If we will talk about the catabolism, the larger molecule will be converted into the smaller molecule. It is known as the glycogenolysis. The breakdown of glycogen, it is known as catabolism. If talk about the anabolism, is known as glycogenesis. If we will talk about the catabolism, it is known as the glycogenolysis, and collectively it is known as the metabolism. So this metabolism is occur. So here is the glycogenesis chemical reaction. Here we will talk about. First of all, the sugar will be enter into the liver or muscle that is used by the exokinase enzyme in the presence of ATP using to produce ADP, and that's convert into the glucose 6 phosphate. It means the carbon number 6 contain a phosphate group in with the sugar. So that's next is the phosphoglucomutase enzyme used to phosphoglucomutase. Mutation means the changing. It means the carbon number 1 is receive the phosphate group instead of the carbon number 6. So that's go into the carbon number 1. It is known as the glucose 1 phosphate. So remember about this. After all, here is the UTP is used. UTP means uridine triphosphate. Uridine triphosphate is like a ATP, and that's attached the uridine instead of adenosine. So uridine which contain a 3 molecule of phosphate group also and ribose sugar, and that's used to 2 phosphate group will be separate, and glucose 1 phosphate uridylyl transferase enzyme used to produce the UDP glucose. UDP glucose mean the uridine diphosphate glucose. So their structure will be here. We will discuss about that, the structure of the UDP glucose. So remember about this UDP glucose is the precursor for the synthesis of glycogenesis. So here is the G1 phosphate uridylyl transferase enzyme used to make a UDP, that is known as the UDP glucose. After all, here is the UDP glucose at last will be formed, and thus made up of uridine glucose and 2-phosphate group, as well as the one more glucose is looking like here. And that UDP will be released and glucose will be separated by the presence of glycogen synthase enzyme. So this glycogen synthase enzyme is attached. Here I draw the structure in the construction formation or anabolic formation. So this anabolism is the polymerization of first few sugar molecules by the glycogenin. So this protein is the glycogenin, which they are used for the first 3 to 5 polymer smaller monomer, and this monomer will be a primer glycogenin, and that's after the using to make a smaller chain to release it. And the next will be, and this bond is the chain, produced chain is known as the alpha 1 to 6 glycosidic bond. It means the carbon number 1 and carbon number 6 used for the glycosidic bond is glucose bond. So there's the polymerization of first few sugar molecules by the glycogenin. So the glycogenin is used for the first few sugars molecule. After all, secondary enzyme will be used. The secondary enzyme is known as the glucoside transferase enzyme. Transferase means the transfer of the sugar into the polypeptide. So here is the second enzyme which they are used to further investigation of the glycogenesis. So these glycogenesis will be produce the sugar. Here is the, you can say that here is the water will be released. So that is known as the dehydrolyzation. So that water will be released to make a glycosidic bond, and this one by one is attached by in the presence of secondary enzyme is known as glycoside transferase. So here is the chain formation will be occur when two chain will come together to alpha 4 to 1 glycosidic bond, that is binding site will be occur, and here is the after the branching release for another site. So these enzyme branching binding site, the enzyme branching enzyme is released to make another branches, and this will be one by one is going on. So here is the structure of the glycogen formation after the different anabolic characters, anabolic activity to produce. Here is the so here is the non-reducing end. Non-reducing end mean the glucose can more add. So non-reducing end is the too much, and one is the reducing end. It is the initial phase. The one is the when beginning of the glycogenesis, the one reducing end will be formed, and that's its reducing end, and it is the branching point. Here is the branching point is also present by the using of enzyme is known as a branching enzyme. So the non-reducing end is recognized to further branching of the glycogen. So the glycogen is a higher density and higher molecular weight is known as the macromolecule, and it is known as the homopolysaccharide. Homo means same, poly means many units, saccharon means sugar. So the here is the reducing end and non-reducing end. Basically exit or add molecules from the non-reducing end because hydroxyl group are present. On the other hand, the glucose will not bind because oxygen is bind, and that's the reducing end. So the only one reducing end per molecule it will be formed. So these show the color of red color in the iodine solution, and that is known as glycogen. So this is the glycogen formation. Next video we will talk about the glyconeogenesis, the breakdown of the glycogen. So thanks for watching. Please subscribe the channel. Okay, bye.